I'm Jen Cannell, and this is Researchers Under the Scope, a special episode we're excited to share with you. Our listeners, our guests know these days, it can be really challenging for researchers to find funding. There's a lot of competition for health and biomedical research dollars, so being awarded a grant, incredibly tough these days. That's where I think at one point there were fewer than 10% of applicants in Canada who succeeded on their first try. This is where organizations like Heart and Stroke come in. And they collect private donations, they support research related especially to heart health and brain health. Now, those donations are often from people whose loved ones have cardiac problems, they've had heart attacks, they've had strokes, and those donations power research. So we're here today to celebrate that research, and not one, not two, but three members of the University of Saskatchewan's Department of Anatomy, Physiology, and Pharmacology. They've been the successful recipients of these national awards. So to tell us more, I want to introduce these three principal investigator recipients. Scott Widenmaier, you are funded for a 3-year grant. We have Changting Xiao and Michelle Collins. They have received new National Investigator Awards. All three of them are assistant professors in the Department of Anatomy, Physiology, and Pharmacology. We're just going to call that APP for short. So hello and welcome, all three of you.
Thank you for inviting us to do this.
Also joining us to help set the stage here is Carolyn Sear. She is Heart and Stroke's Director of Health Policy and Systems here in Saskatchewan. Thank you for being here today, Carolyn. What can you tell us about these National Heart and Stroke grants?
Yeah, so maybe a bit more background context about heart and stroke. We've been leading advances in heart and brain health research for more than 70 years. Our mission as an organization is to promote health, save lives, and enhance recovery for people living with heart disease and stroke. And one of the ways we are able to do that is through supporting research like Scott, Michelle, and Changting, who are here with us today.
How rare is it, honestly, that you would have three biomedical researchers from the same department, same institution, all winning these grants at once?
Yeah, I think it's definitely something to celebrate, and here in Saskatchewan, something to be really proud of, that we are so well represented in our national awards. At Heart and Stroke, all of our awards are administered nationally, and there's a gold standard peer review process for those to be awarded. So in order to receive funding, applicants really need to rise to the top and receive a really high rating. And it's a testament to the excellent research that they're doing and the high caliber of their applications that we have three researchers from the same department who were able to be funded, and I'd like to congratulate you all on your success.
Thank you.
Well, and it's funny because there really is a real human side to the people who are behind these donations and behind these grants. And this is where I'd like to share one of those perspectives with you. I did have a chance to sit down with one heart patient here in the Saskatoon area. She's a mother to 4 children. She's a teacher. Have a listen.
My name is Meredith Rhinas, and if we bumped into each other somewhere, you might know me as a high school teacher in Warman, Saskatchewan. You might know me from my presence on social media. I run a Instagram account on lifestyle and get a chance to talk about all kinds of things involving motherhood and heart health and just all kinds of things. So I was actually diagnosed with bicuspid aortic stenosis when I was about 18 months. So I have always had a congenital heart defect. I had a sort of non-invasive procedure when I was 10, where they put a little balloon up into the aortic valve to stretch it out and gave me a little bit more longevity in terms of being able to be active and do day-to-day things without running out of breath. So that did improve my quality of life quite a bit in my teenage years. And we always knew that one day I was going to need surgery to fix it. But it became really problematic for me as I aged out of pediatric cardiology and was kind of transferred over into adult cardiology. And then once I began to have babies, things got really complicated for me. I had 2 children in my 20s, and those pregnancies were not very difficult. I was able to have them both naturally. I felt good, very minimal cardiac presence. And it was with my second 2 pregnancies in my early 30s where things really kind of went off the rails. I was quite ill with my third. I had a lot of difficulty breathing. It just felt like a lot of pressure on my chest. I was very swollen, like my hands and feet were incredibly swollen. And a lot of these things, like they are symptoms of pregnancy also, right? I was exhausted all the time, but I was like, well, I'm pregnant and I have 2 other children and I work full time. So you just sort of go, I'm just pregnant and I'm in my 30s and I guess I'm out of shape. And, all of these things you just think. And that pregnancy was, well, it was very difficult and it ended the whole thing was very difficult. When I was 4 months pregnant with Nora, she's the youngest, I got a phone call saying that my cardiologist had stepped away from medicine indefinitely and I needed to find a new cardiologist. And so when I wound up meeting with the new cardiologist, he basically told me that things had progressed and were very severe. They were very concerned that I was going to basically wind up in congestive heart failure, like fully. And that kind of rocked me like a lot because I just never considered a scenario where like my baby might not survive. Do you know what I mean? Like I thought it would be worse for me and that, I was going to end up having to have surgery or something right on the table. And so I was very, very lucky in that particular situation. I did have to obviously recover from that C-section. Nora spent about a month in the NICU. And then 3 months later, I was scheduled for the open heart surgery where they replaced my aortic valve with a mechanic one. That did also not go well. And the reason it didn't go well is also because I'm a woman and the metal valve that they actually put into my aorta was a little bit too big. It wasn't built for someone as small as me. And so not that I'm so small, but the surgery I had is typically, men 65 plus and much larger, and it resulted in some complications. And it's no one's fault. It's just that I'm very, very lucky to have survived. So yeah, those of us who were born with congenital heart defects, the incredible piece is that the research has come along enough that we're living. Like we are not, dying as children. And that's kind of a very blunt way to put it. But now, there's all these procedures and these surgeries that are keeping us alive, kept me alive as a child. But now I'm an adult and I'm a young adult who's living with heart disease, and the research hasn't caught up for, moms who are having these babies who have these heart conditions. And I think that that's a piece that is missing. I do think there's a missing link there, right? And so I want people to be able to learn from my story and my situation. There were so many questions I had as a woman post-surgery, and I was like, I don't know if this is normal. Like, all my hair is falling out, or, is this connected to the surgery? All of these questions, and finding those other women online helped to alleviate some of those worries and help to build some community. And you have to be able to own that this is your body and your experience, and that you deserve to know and understand and really understand that your questions aren't a problem and you aren't being dramatic. And, all of those pieces I think are really important for women in particular when it comes to advocating for our own health.
That's Meredith Rhinas. And as a baby, as you heard, she was diagnosed with bicuspid aortic stenosis. And yeah, in her words, she wants the research to catch up. So just listening to her, Scott, Michelle, Changting, what runs through your mind?
It's a really sad story, how things turn out, and I'm glad Meredith is great and she's moving forward with that. And for me, I think it's motivating. It motivates us to want to pursue research and find new answers for heart and stroke conditions.
Yeah, I find her story very powerful because going through pregnancy already is a very scary time, let alone when you're dealing with health conditions that you don't really know how to navigate. And I think it also speaks to the fact that 1% of babies born in Canada have a congenital heart defect. And with the advances in research, like Meredith had said, 85% of these kids are making it to adulthood. But it does mean that there's a very particular subset of patients, these adults with congenital heart diseases, where the research maybe is lacking. So for instance, the heart failure that she mentioned, actually patients with heart failure as a result of congenital heart disease have a different type of heart failure than the regular variety of heart failure that happens. And often these surgeries that they receive as children can lead to things like cardiac arrhythmias or irregular heartbeats, which are something that has to be dealt with as the individual ages and deals with other sorts of complications. So I think it really speaks to the gap in research that's there.
Yeah, just wanting her questions answered. And Changting, what did you think just listening to her story?
So Meredith's story speaks a lot about the life quality affected by heart disease and strokes, and that also speaks about the need that we, from a research side, what we need to do more to find out the cure and to help with the patients with heart disease and strokes.
Yeah, it's funny. There's so much that goes into it. Carolyn, just listening back to Meredith's story, for you, what rang really true?
I've been able to hear Meredith's story now a few times. There's always something new that comes out of that. And I think I really value having that human face behind these conditions. And as Michelle stated, everything's so interrelated and connected. I know Meredith shared with me in the past, she has to teach her kids the signs of stroke, or fast signs of stroke, because she's at increased risk of stroke as well. So she may have congenital heart disease, but it's connected to heart failure, it's connected to stroke, it's connected to arrhythmias. And then it just really points to some challenges when it comes to women and inequities in health and outcomes and treatment and care as well. So there's a lot there, a lot to unpack and look into for sure.
Well, and I know that initially when I was learning about heart and stroke, I wouldn't have thought the gut was actually part of it. I'm like, well, there's my brain and there's my heart. But Changting, you have looked really closely at things that our gut is doing. It's sort of the second brain, you told me when you were last on the podcast. What are you studying now?
We are studying in several aspects of what the gut does. One particular aspect is how the gut handles lipids, because we know that lipid disorder is a major risk factor for people with diabetes and obesity to develop heart disease and strokes. So we're looking into that.
And can you tell me, in terms of bringing this work into the laboratory and some of the grant that you're working with, what does this let you do now? Like, what are you looking at when you're actually in the lab and your team is?
So we have several projects going on in the lab. And first of all, as I just mentioned, we study how the gut handles fat. And we know that the fats that we eat through our food are digested and absorbed into the absorptive cells that are called enterocytes. So we want to understand how these enterocytes process the fat inside of the cells. Some research have been done, but still there's a lot new to us, and there are new metabolic pathways, there are new cell organelles that are identified to play a role in this. So we want to look deeper into this and see how that happens and what are the control points. So that's one aspect of the research.
And in terms of, I know that some of our listeners will go, well, how does that apply in later life? But I'm starting to put it together. What's happening in my gut does affect my risk of heart and stroke. How can you see this working in the future?
We have several projects just looking into how the gut works and how the gut handles fat, and we're looking at how fats are processed in the gut absorptive cells, which are called enterocytes, and we are looking at new metabolic pathways and new cell organelles in the cells, and to see how they participate in fat handling. We want to find the control points. So that's one project. The other project is that we now think the gut is a fat storage organ. So the fat that we eat from our food, they do not necessarily immediately leave the gut after they are processed. So the gut can actually store some of the fat in the gut for quite a long time, and that actually relates to certain diseases. So we want to find out how that happens and what we can do about it. But we are also looking at the non-fats. We're not just working on looking at the fat, we also look at what the gut produces, other factors. The gut is very powerful organ. It does a lot of things. It processes food, it processes fat, but also processes other nutrients and secretes a lot of gut hormones. If you are aware of the latest drugs for diabetes and for weight loss, those actually are discovered and developed from studying gut hormones. So we want to look at the gut and see what else does the gut secrete or produce, and what can we learn from that, and what can we use that knowledge for for our human health. A final project is that we are testing several novel drugs and see how these drugs affect the gut functions and affect the gut handles lipids and non-lipids. So these are all the active projects that we are working very hard in the lab.
That is a lot. Scott, Michelle, as he speaks here, anything you'd like to weigh in with? Like that's just in terms of the kind of work that Changting is doing this sounds like something I don't hear about all that often.
Yeah, I think it reflects maybe an underappreciated aspect of the basic research that we're doing is a lot of the major discoveries happen decades before it turns into a clinical translational outcome. For instance, the gut hormones he talked about were discovered in the 70s and 1980s, and then it took at least 2 decades before the first version of the drug was made to treat patients in the early 2000s, and then Ozempic came on the market like a few years ago. So that's 50 years later before this really turned into a powerful therapy. And then the first hormones ever discovered were discovered from the gut. They didn't know that hormones existed. So the gut has a lot of places and a strong future for new therapies, I think.
Yeah, I'd also add in that I think Changting's lab is using some really innovative and exciting approaches to study this question. And he's got a very unique skill set to really tackle these problems. So it's exciting to see what his lab will bring forward in the future.
And in terms of the future, what do you see as a possibility?
Our lab, as Michelle mentioned, that we use some special tools, and actually we have a unique technique that allows us to provide nutrients on one side of the gut and collect and see what comes out of the gut from the other side of the gut. So that is a very unique tool, and we do all these in live animals. We are one of the very few labs in the world that are capable of doing this. So if you walk into our lab, you'll probably find us working like a surgeon sometimes. So that's like the fascinating part. I tell the students when I teach in my class, I show them pictures about us putting gowns like surgeons, and that fascinates the students too. So on top of that tool, we also use other techniques like biochemistry, molecular biology, and imaging, etc., to look at what happens to fat and to non-fats at the body level and at the organ level, the gut as our organ, and on the cell level, and also inside of the cells. So that's really makes us capable of looking at answering questions from all different levels.
And just in terms of where you can see this helping heart and stroke foundation patients maybe down the road or in the future, what do you see there?
So by looking deeper into the gut cells, we hope to find druggable targets so that we can develop new drugs to target the drug and to curb the lipid output from the gut. So that will correct lipid disorders and fight against heart and stroke disease. And we also hope to understand how certain drugs work. And then by looking at what the gut produces, fats and non-fats, we hope to understand better what the gut actually does, what else the gut does, and what we can do at the gut level. The Greek physician Hippocrates once said that all disease starts in the gut. I believe that's true, and maybe that's my bias, but I think by studying the gut, we have the opportunities to say that we can target the gut and we can stop all disease starting at the gut. So, sufficient funding for research will definitely help us to reach that goal much faster.
Now, looking at it, I know, Scott, the last time we chatted, it was largely about cholesterol and sort of the little fires that it can light and the fuel we take in, like right down to the molecular level. These days, I know you're studying the liver, and not just the liver itself, but also the way it handles stress. With this grant from Heart and Stroke, what question are you exploring?
Yeah, thanks for the question. I would say that I have my own biases. So the liver is, maybe you could consider it as sort of the Amazon of our body. So when things get through the gut and then they get into the bloodstream, the liver is the center point at which all the packages come. And then the liver decides what needs to go where, and it reprocesses things in different ways, and it has the ability to recognize things that are not supposed to be there. So for instance, to come back to physiology, if bacteria leak through the gut, the liver is the first place to catch those things and get rid of them. If the things that shouldn't be in the body at all, the liver is the first place to catch those things. But this can be a very stressful process because a lot of things that come through can cause damage. It can cause little micro damage at the tissue level. And so the liver is equipped with a number of different factors to help fight against that stress. And my lab is spending a lot of time trying to understand how those stress factors work at the molecular level, how they fight the kinds of stress that we see in obesity, in dyslipidemias, even in infection, because all those things can precipitate to a heart disease, can precipitate to stroke. Actually, one of the major things that happens after a major inflammatory event is you have a lot of clotting, a major risk factor for having major problems later. So the liver can deal with all those aspects, and we're trying to understand how stress defense programming, can fight those things.
I know we talked about being right down to the molecular level. What transcription factors, what genes are you looking at these days?
Yes, thank you. Well, the major ones that we're focused on right now, their names are nuclear factor erythroid 2 related factor 1 and 2.
Catchy.
But you can, exactly. We like to call them NRF1 and NRF2. And they have been identified to be expressed in all tissues, but we're really studying them in the liver and trying to understand when we take those genes out of the liver, how does the body deal with the types of stress that are linked to heart and stroke disease? When we add them back to the liver, or when we make them work better in the liver, how does that affect the ability of the liver to deal with dyslipidemia or infection, that kind of stuff?
So seeing how it works in times of disease and if you're trying to rebalance it out in times of healthiness, like both sides.
Yeah, and the real struggle for us is sometimes we can get really strong results that are very exciting, but it can sometimes be in a place that's not exactly translational. And so we're working really hard to develop model systems that help us study whether if we manipulate these genes in the right context, is that actually realistic now? Can we target these genes to treat something that's important for heart and stroke disease? So
Okay.
that's what we do in the lab.
So yeah, like down the road, I think that's kind of what you're pointing to. Where can you see this leading us, especially when it's combined with research like what Changting is doing?
Well, my hope is that NRF1 and NRF2 can be targeted with powerful therapeutics, and that we can then target them in the right context, and patients with a variety of conditions that are linked to heart and stroke disease can be treated and have better lives.
Yeah, Michelle, Changting, is there anything that kind of caught your ear as Scott was speaking, or anything you'd like to weigh in on his research?
Yeah, I think I would add in that, as Scott had mentioned, NRF1 and 2 are expressed in other tissues of the body. And so applying the lessons that Scott can pick up from the liver to other tissues might shed some interesting insight on other aspects of organ biology and organ physiology.
Yeah, Scott really explained how the liver, how important is the liver for health, and he didn't mention, but he's doing the work. I think, if I can. So the liver is very important in terms of generating lipid particles and sending fats into the bloodstream. And actually, lipid disorder is very closely related to the liver. And actually, what Scott is looking into, some of the genes actually affect lipid secretion from the liver. It affects, it produces the bad cholesterol, and it also produces the good cholesterol. So by manipulating the nuclear factors, the genes in the liver, I think there's a great potential to actually help correcting lipid disorders through Scott's research.
Can I add as well, Jen, as very much a non-researcher myself, I think I often incorrectly think of, if somebody's researching and studying heart disease and stroke, we're looking at the heart, we're looking at the brain. And so it's really neat to hear about people who are studying the liver, who are studying the gut, and the potential applications to prevention and better management of these conditions in the future.
I didn't realize how many systems they help regulate. Mm-hmm. Like, it's really incredible what happens through that whole digestive tract. And I'm the same way. I didn't really associate it with heart and stroke, but here we are. Now, Michelle, I've always associated your research with the heart. I know we talked about zebrafish hearts the last. Actually, it was really cool. She took me inside, like, got to see the zebrafish themselves. And then she showed me how, was it the channels, like where things were running and how you can actually look so up close at things.
Right, we were looking at how calcium ions move through the heart.
Yes, it was really cool. And I'm looking at it now, and in terms of on like sort of the genetic side of this and on the way hearts develop, what are you able to look at now?
Yeah, so right now the lab is still looking at those questions that we talked about in our last podcast. So, broadly speaking, my lab asks the question, what makes the heart beat? We do this from two different approaches. We have sort of a fundamental natural sciences angle where we try to figure out how electrical signals and mechanical muscle contraction are combined, and this happens through the movement of calcium ions, or that's where we're focusing at least. We are taking different strategies to look at this. So for example, using zebrafish, we can look at the movement of calcium ions and study how they respond in different contexts. On the other side of the lab, we're now studying, well, we've also been studying cardiac arrhythmias. So these are disorders where the heart beats irregularly. So either too quickly, too slowly, or with an arrhythmic pattern. So my lab has had a long-standing interest in a common cardiac arrhythmia called atrial fibrillation, or AF. And so what has been it's been historically thought that AF is really an electrical disease, so by problems with ion channels. But what we're realizing now from a lot of different genetic sequencing studies is that there's a significant component or a significant contribution from genes that are really early transcription factors that build the heart during development. And in fact, the heart is the first organ that forms in the embryo. In humans, this tissue starts forming at 4 weeks of development. So this is often a time when women or pregnant people don't even know that the pregnancy is there, right? So it's very susceptible to perturbations. But so we're using a zebrafish model that lacks this transcription factor. It develops symptoms of atrial fibrillation, and we're using that to understand what are the initial triggers and factors that drive disease initiation and progression. And more recently, my lab has started collaborating with a pediatric cardiologist at the Jim Pattison Children's Hospital, Dr. Teresa Pocket. And we're also collaborating with the medical genetics team over there, led by Dr. Bita Hashimi. And our goal together is to try to identify novel genetic variants in pediatric patients that have neonatal cardiac arrhythmias. And so to do this type of work, we're recruiting families into these studies. We're doing genomics research to try to identify novel variants that are in genes that might be linked to the disease, and then do functional genomics approaches to understand, what is the consequence of that variant, or what do these genes do in the normal and the diseased heart.
Oh, wow. And so in terms of like the lab work itself, I know the zebrafish are definitely a heavy part of it, but what does your lab work look like these days?
Right, so we do still do a lot of zebrafish work. So there's, students doing a lot of live imaging of zebrafish. So when you visited, we looked at the confocal microscope that we have.
It's so cool because it works like in colors, and you can set it to like, here's what I want to see.
Yeah, no, it's a really powerful machine that we're super fortunate to have here. So a lot of the lab does that kind of work. Now we're getting more into different cell models, so using rat and mouse cardiac cell lines. And we're starting to develop induced human pluripotent stem cell models, which really helps us tackle this issue of novel genetic variants in humans. But on a day-to-day basis, so at least for the pediatric part of our work, we've had some pretty exciting developments in that story. So we've been able to recruit patients and their families to the study. We've done some whole exome sequencing of one of these families, so from an affected child and an unaffected parent. And right now we're doing a lot of bioinformatics work to try to figure out if there's a genetic variant that is perhaps linked to this neonatal cardiac arrhythmia that this child experiences. And we're in the process of developing disease models, both in cells and in zebrafish, to try to model what the effect of this variant is in an in vivo or in vitro system.
To actually see it happening in real time.
Yeah.
What goes through your mind just listening to Michelle?
Oh, this is really great work. I think what strikes to me is that your link from the basic fundamental research in the lab and the clinics, and by looking at both zebrafish and cells and then human babies, right, to tackle this genetic cardiac congenital heart disease. So that's really nice work. Yeah.
Yeah, I think it's very exciting work, and I love how it has a Saskatchewan-specific spin to it. These are going to be mutations in Saskatchewan families. It might be the mutations in a very small cohort. It might be mutations that are broad and globally affecting neonatal patients. And it reminds me of how some of the most important advances in cardiovascular research was done doing the same kind of research. So the discovery of the LDL receptor, which is important for getting rid of that bad cholesterol in the blood, that was identified in patients that they just called at that time familial hypercholesterolemia. So they had dramatically high levels of cholesterol in their blood. Sometimes it almost looked milky white, but they had no idea why, and it took time for genetics technology to catch up, and finally they found the mutations in this one particular molecule, and that led to some really important studies and the development of statins, which were used for now 50 some years, and we're looking now for the next generation, the next version of that through this kind of work.
Yeah, and I think that, just to add on to that, I think the idea of having a model where you can begin to do therapeutic or directed testing is really the next step of where we'd like to take this. So if we can create an animal or a cell model that carries a patient-specific mutation, then you can start to use this in some sort of drug screening platform to try to identify compounds that might be I guess more beneficial for certain individuals based on their genetic makeup, and that's sort of within the realm of personalized medicine that would be very exciting to take this work in the future.
Geez, Carolyn, just listening to this,
My brain is full.
right? But what does that also tell you just about, like, this is one department, APP, one department at U of S. What does this tell you about what's happening research-wise?
It is fascinating, it is exciting. You know, at Heart and Stroke, we fund different types of research. So at the population health level, health systems, clinical, and then biomedical. And sometimes it's harder for people, and for myself included, to sometimes connect those dots between, you know, what's happening at the biomedical level and what is that potential application. But I can see it listening to you talk, and Scott, you've given lots of examples about how, you know, that biomedical research eventually informs, you know, prevention, clinical management, future drugs that are developed that are really making a difference in so many lives of people. So it's there, and you know, my work at Heart and Stroke, I'm involved more on the health system side, the policy side, but I wouldn't be able to do that without having evidence to inform recommendations and best practices, and that evidence comes from research like this. So it's really exciting to see U of S leading in so many areas and being so well equipped to do that based on the people we have, the technology, the resources, the passion. You know, Changting, you shared that your lab is one of few in the world who is doing and able to do the research that you're doing. So I was just going to say that, you know, the work that all three of you are doing is such a tangible example of what Heart and Stroke is able to do with the generous donations from many people living across Canada and here in Saskatchewan. And we've made so many advances in research over the last 70 years, but we know that there's still so much to be done, and Meredith's story is such a good example of so many people who are still living with these conditions, and there's still so many unknowns. So very important to continue to support that work to happen.
And it's tough because sometimes you want the research to catch up right now. And Scott, you pointed out, sometimes it takes decades. Like just looking at Ozempic alone took decades from when we started learning about where it would affect to here we are now rolling it out. But for people like Meredith who are watching this and thinking about it, where do you see reasons for hope?
I think from our aspect of work, our goal is to identify genetic variants that might be important or allow for genetic screening of other family members, of other individuals with similar phenotypes, not only in Saskatchewan, but perhaps on a larger scale as well. I think this information is really important to empower patients to know what's going on, and also decide on the best courses of treatment based on their genetic information. And I think that would be, those types of areas of research are really going to be important as the field moves forward, and with the support of Heart and Stroke and other foundations like that we're able to push forward these areas of research.
Well, I think the reason for hope would be that, though I discussed that it took decades and so many years for these discoveries to happen, that was at a time when technology was nowhere near what it is today. And so now, another example would be the discovery of PCSK9, which is, you know, just as a gene, but it plays an important role in the levels of bad cholesterol. And that the cloning of that gene, the linking of that gene to the disease, and then the discovery of, and then the development of a therapy that treats that disease happened very fast, within less than 10 years. So now things are happening way quicker, way faster, and even the COVID vaccine took very little time to develop as compared to how long it's taken in the past. So the technology's there, the ability to find the genetic variants are there, and these things can now move at a much faster pace. So I think there's hope in that.
So as a biomedical researcher, we tackle questions, we tackle, we look for fundamental biology, but we always have the mission of addressing health needs, to fight against a certain disease. It's like our mission. The primary mission is to fight against heart and stroke disease. So I've been working with basic scientists and with clinician scientists, and we look at both the bench side and the bedside. So you can see that the every small step that we move forward looks small, but in the long run, we accumulate knowledge. We just build up what we make each small step, each little discovery. We build on that and we move forward. And then at a certain point, we will see the big jump, a big leap for example, the latest drugs for diabetes and weight loss, and they actually have been shown to actually benefit the heart a lot. They all have cardiovascular benefits. So that's something that not so many people had thought about, but you can see that through the years, through the past few decades, all the research now actually bring the benefits to the people to benefit their hearts.
And I think another reason for hope, since 1952, which is when heart and stroke was established, there's been a 75% decline in mortality rates for heart disease and stroke. So Meredith pointed to this in her story about the reason she's kind of facing some of these challenges as an adult is because research and treatment and medicine has allowed her and others like her to live past childhood and into adulthood. And there's so many other people with congenital heart disease. So that's what we've been able to accomplish in 70 years, and just look at the potential for what we're able to do in the next 70 years through the research that's being done.
There are so many reasons for hope. Thank you all for being here and for talking about them with me today. I really appreciate it.
Yeah, thanks for having us.
Thanks for having us.
Thanks so much.
Well, Michelle Collins is an assistant professor of anatomy, physiology, and pharmacology who specializes in the molecular basis of cardiac development and function, the building blocks behind congenital heart defects. Thank you so much, Michelle.
Yeah, this is great. Thank you for having me.
And Dr. Changting Xiao is an assistant professor in the same department, and he specializes in our physiology, our metabolism, the gut. He and his team really study how the body handles those lipids, those fats, in both health and disease. So thank you so much for being here with us, Changting.
Thank you for having me here.
And Dr. Scott Widenmaier is an assistant professor of anatomy, physiology, and pharmacology. Yep, same department. Using that genetic technology, molecular biology, and systems biology, he's studying the pathway, the defense systems that stop the liver from getting sick and stressed. It's the way the liver defends itself against inflammation and all that metabolic stress. So, Scott, thank you again for being here.
Glad to be here.
And Carolyn Sear, the Director of Health Policy and Systems for Heart and Stroke here in Saskatchewan. Thank you so much for being here on the podcast.
My pleasure. Thanks for having me.
Well, to find out more about what Scott, Michelle, and Changting are working on, you can go to medicine.usask.ca. You can also find some more links there that take you right to the University of Saskatchewan's graduate programs in anatomy, physiology, and pharmacology. This research we're talking about today, it's supported largely by Heart and Stroke. You can see more about them at Heart and Stroke, all one word, heartandstroke.ca. Researchers Under the Scope is a presentation of the Office of the Vice Dean of Research at the U of S College of Medicine. We record this podcast on Treaty 6 territory, and as such, we pay our respects to the First Nations and the Métis ancestors of this place, and we reaffirm our relationship with each other. We'd also like to thank you for tuning in and for listening. I'm Jen Cannell, I'm your host, and if you find those 3 little dots in the corner of your podcast app, go hit follow, or better yet, go online, tell a friend about this episode. Biomedical research really does matter, and we are so grateful to you for sharing the love here on Researchers Under the Scope.